So hello everyone, my name is Patrik Engström. I'm a principal scientist at Primrose Bio. So today I will talk about double-strand RNA formation and how RNA polymerase choice can reduce it. So double-strand RNA is an unwanted byproduct in vitro transcription reaction that can be costly to remove downstream. So in in vitro transcription reactions, we have a DNA template, we have normal or cap analog, we have nucleotides, we have a buffer system, and of course we have an RNA polymerase that is responsible for generating transcripts. And in most cases, the transcripts are full length, but there will also be most often be a subpopulation of transcripts that are these abortive short transcripts. These different RNA species can stimulate double-strand RNA generation. There's many different mechanisms described out in the literature, and one of the most common one is predicted to be this loopback double-strand RNA, where we have a 3' extension, and we basically have the RNA polymerase transcribing back on the intended full-length transcript, forming double-strand RNA. Another proposed mechanism is priming of the full-length transcript with this abortive transcript, stimulating also transcribing back on the full-length transcript, forming this double-strand RNA. So there are many different ways to detect and quantify double-strand RNA. The most common one is sandwich ELISA, which contain two different anti-double-strand RNA antibodies. One is used to capture the double-strand RNA in the samples, and one is used for detection either via HRP conjugated or fluorescent conjugated antibodies. And here we have an example of where we had measured double-stranded RNA per total mRNA generated by T7 in black. And you can see here we have tested how much double-strand RNA is generated in different in vitro transcription reaction with different amount of DNA template. And we can see there's quite a bit of double-strand RNA generated by T7, but with these non-T7 enzymes in orange that we are working a lot with in our lab, we can see that regardless of the amount of DNA template, we have very little double-strand RNA generated. So these non-T7 enzymes are, of course, of high interest, but there are some caveats with those that I will walk through here, and also a solution on the caveats of those. So here we are looking at all the four main KPIs. So we're looking at double-strand RNA, capping efficiency, yield, and integrity. And here in black, we have put T7 RNA polymerase to 100%. So then when we look at one of these non-T7 RNA polymerase enzymes, again, we can see that the double-strand RNA levels are quite low, up to 10-fold lower than for T7. For capping efficiency, we can also see in this experiment that it's higher than T7, as well as integrity is also higher. However, for this non-T7 RNA polymerase, we see that there's a trade-off. We see that the yield is quite low. So what we did is we took this non-T7 RNA polymerase and also other non-T7 RNA polymerases, and we exposed them to our evolutional platform and protein engineering platform to see if we can improve on, for example, here in terms of yield. So the outcome of that was that we identified an engineered non-T7 enzyme that now has no trade-offs, and the overall performance is excellent. So that gives you an example of starting with a non-T7 RNA polymerase, engineer it to make it an overall high-performing enzyme is a viable path to generate an enzyme that can be used in mRNA manufacturing. So just to summarize that again, so yeah, we have identified non-T7 enzymes that reduce double-strand RNA significantly, but they show trade-offs. But we have used those enzymes as a good starting point for further improvement. And the final products show great activities with overall performance that's really high with no trade-offs. Thank you, everyone.